Morning, everyone. I would like to first thank the Magnus Group for inviting me to present my work here. I work at NUS Environmental Research Institute, and there we are mainly working on plant microbes interaction, also urban farming, but the work I'm going to present here is different. I'm going to present on promoter architecture and transcriptional regulation of genes, upregulated in germination and coleoptile elongation of diverse rice genotypes. Those are tolerant to submergence. Okay. We all know that rice is one of the major food crop of the world and is the major food crop in Southeast Asia. It's produced worldwide. And also there is, increasing gradual production since Green Revolution. Now, of course, we have super Green Revolution and rice production has really increased a lot. But the problem is there is prediction that by 2050, the population will increase to 9 billion. Then how to feed all these people? And even if the rice production is increasing, it's the productivity is affected by different abiotic and biotic stress like light, drought, flooding, salt, minerals, and also different types of bacterial fungal disease. So it's quite important to characterize the stress response mechanism and to improve the productivity of rice and of course the other crops. And here I'm going to talk about the submergence stress. We all know that in Southeast Asia, we grow rice during rainy season. And that time the rice field is flooded. And normally people go for direct seeding because it's the manual labor is cheaper. And in Southeast Asia, many farmers are poor. So they prefer the direct seeding compared to the transplantation. So what happens, the fields are flooded. But the rice seeds can germinate. And rice is the only plant, the unique plant that can germinate and elongate its coleoptile during the submergence condition. What happens during submergence? Sometimes, depending on the water level, it can be hypoxic or anoxic. Anoxic is without oxygen. Hypoxic, there is less percentage of oxygen. So, but even if some of the rice varieties are tolerant to anoxic germination, the rate of germination of the coleoptile, the rate of germination and coleoptile elongation varies from genotype to genotype. And to study that, to elucidate the unique transcriptional regulatory mechanism that regulates the difference in coleoptile elongation, we wanted to do the promoter architecture analysis because these are due to either metabolic or transcriptional regulation. So to do that, we did some, an ab initio approach to reveal the promoter architecture and possible transcriptional regulation of rice genotypes from different background, different diverse background. And we believe that the information that we derive, that's of course in silico analysis, can potentially guide in developing new varieties for direct seeding. The aim of doing promoter analysis is it can provide information on the associated binding sites with the cognate transcription factors. And also it provides useful information between hormonal regulation, different hormonal signaling, and because the hormones, they are involved in regulating the downstream gene through interaction with other regulatory molecules of the signaling pathways. So to do this, long ago, this paper was published in 2012. So we did the work sometime 2010. The rice genome was sequenced 2005. So the genomic data was available, but it was still, lacking many information. So what we did, we did the work on a Japonica variety Nippon Bare. Nippon Bare is very famous for germinating under anoxia. We have the many indica varieties, but indica varieties, many are also sensitive. But this Japonica variety is quite well known. So we took the microarray data of the gene expression data of upregulated genes, and then we did the promoter architecture analysis. And this is the method we followed because I was working in a bioinformatics lab at that time. So we extracted our own promoter sequences, that is minus 1000 and plus 200 nucleotide relative to TSS. Then we used Dragon multi-builder program. This program was also developed by our lab. So we used our own program. And then we did the motif analysis. Then we find the motif or binding sites. Then we look for their, what are those motifs in different databases like Transpac, Place, AGRIS. Also, there is another plant database like Plant Care. But Plant Care has very little information and it's quite old. AGRIS based on Arabidopsis. Place also since 2005-06 they have stopped curating. So the best one is Transpac, but then you have to pay a lot of money to use that database. Anyway, we looked at the putative binding sites, verified in these databases, and then we linked them with we found the associated transcription factors, link them to the upregulation of those transcription factors. And then we believe that those transcription factors could be involved in the regulation of the coleoptile elongation. So this data shows, that time not much information was available because genome was just sequenced 2005. So we could find some transcription factor binding sites. Those are associated with MIB transcription factor. Then ARF, that is auxin responsive factor. Then MIB, this MIB is GA responsive, that is gibberellic acid responsive. And this MIB is ABA responsive. Also, we found many BGIFs. And those are, some of them are ABA responsive, that is abscisic acid, and also ERF, ethylene responsive transcription factors. Then we were really surprised to find the ABA responsive transcription factor. Because what happens during seed germination, when there is imbibition, the ABA level goes down because during seed maturation, there is a ABA level goes up. And when there is imbibition germination, ABA level goes down, GA, gibberellic acid goes up, and that induces the alpha amylase gene to break down starch to sugar, which is required for the germination and elongation. So we were really shocked to find many binding sites. Those are ABA responsive. And when we published this paper, we got a lot of criticism that how come, you have ABA responsive transcription factors. And then after many years, I was working in an US chemical engineering lab, and there we wanted to develop the rice genome scale model. And what we did, because the genome was sequenced, we had many microarray data. Sorry. And then at that time, microarray, now gene expression data, transcriptomic data. Then we have proteomics data, we have metabolomic data, we have vast information on all these data. Then how do we get the information from this? How do we get combine these data to get the knowledge? That can be done through system biology approach, and that is through mathematical modeling. Because I was in the chemical engineering, so they were more interested in modeling. And so there are different types of models. First one is interaction type. That is, if people are using some genomics, some people are involved, expert in genomics, they may be knowing. The example is rice cycle, where all the pathways are connected to each other, but you cannot have the flux flowing through the pathway. So it just, this type only give you information about the pathways. And the constant based is here you can simulate to get the flux through the pathways. Third one is the most comprehensive type, but then it lacks required experimental parameters. So we use the constant based modeling to develop our rice model. Initially, what we did, we developed two core model. one leaf specific, one root specific. But then ours is seed germination. So we are interested in the root specific model. To develop this model, it's a lot of manual work, a lot of curation and thing. So like we followed 3 steps: step 1, step 2, 3. In the first step, we collected data from different databases regarding the pathways, because each plant has like many pathways and reactions, and whether different. These are all metabolic reactions. And in the second step, we did the manual refinement, because in plant cell, it's very important to put the right reaction in the right compartment, because we have mitochondria for respiratory enzymes, we have chloroplast for photosynthesis. So we had to manually curate that. And then also look at the the gene protein reaction, that is important. Also the reaction reversibility, some are unidirectional, some are reverse reaction. That is also quite important for the flow of the flux. And then third one is model evaluation, because unless you experimentally validate, you cannot be sure that whatever you are doing is right or not. So third one, we did model evaluation using our leaf specific data. We did some work on drought stress. I'm not showing here. And we found that our model evaluation and the experimental data, they're very close to each other. And for the seed specific also we did. For this, what we did, we grow the rice suspension cells under anoxic condition, air condition, using sucrose, different types of carbohydrate in the medium. And we found the flux through, the growth using our model is very similar to the experimental data. So, and these core models are having only few compartments like cytosol, plastid, and mitochondria, and not many reactions, because the secondary metabolic pathways are not there. Many other things are not there. Then once we are sure that our model is working, we again did the same work, the Japonica variety Nippon Bare. We wanted to simulate and then compare with the experimental data. What we found, the flux through when we did the seed germination under anoxia, we found the flux through the glycolysis and external fermentation was much higher. That we know that they get energy during anoxic condition. They get energy mainly through alcoholic fermentation, because during that time there is no oxygen, so Krebs cycle is not developed. Rice during germination, people have done some work. They found that the Krebs cycle is truncated, but the main thing is the alcoholic fermentation. So once we are, it was quite matching to the experimental data. Okay. Then what we did, we look at did promoter architecture analysis by taking the important genes like sucrose synthase, pyruvate decarboxylase, alcohol dehydrogenase, and aldehyde dehydrogenase. And interestingly, we found the same transcription factor binding sites common to all these genes. And we look at the upregulation of those transcription factor. It was quite nicely matching with that. So we hypothesized that these transcription factor, BIP, BGIP, and ERF, zinc finger, could be involved in the transcriptional regulation of the rice germination under anoxic condition. And then after a long time, again, I was quite interested to do this. This is a public data. What they did, they used different genotypes, from diverse background. Nippon Bare, they, consider this one as moderately tolerant. And they have two recombinant inbreed lines, F29A and F274-2A. Those are derived from a cross between Nippon Bare and IR62. Nippon Bare is a tolerant variety. IR64 is a sensitive variety. And then they use two natural genotype originated from Southeast Asia, 8391 from Laos and 8753 from Indonesia. So here you can see in the air and anoxia, this is Nippon bare. That's why they think moderately tolerant, because the coleoptile elongation is much less compared to these varieties. And this one is sensitive variety IR64. These two, they think that these are the highly tolerant variety. And these two, even the coleoptile elongation is much longer, and they, these are, according to them, is highly, extremely tolerant varieties. So what we did, we wanted to look at the promoter architecture again to see what is common and what are the transcriptional regulatory mechanism. So we did few sets, like we took all 5 tolerant genotypes, the upregulated genes. Those are common to all 5 tolerant genotypes. Another group, 4 highly tolerant genotypes. So the first one was all 5. Not the sensitive one. Second one was all 4. Third one was these 2 extremely tolerant varieties. And then we had 2 additional analyses to confirm whether our analysis is giving the similar results. We took 2 intermediate tolerant group, that is 2 highly tolerant genotype, these 2, and to compare with the group that has 4 highly tolerant genotypes. And another analysis, 2 genotypes, Nippon bare, that is moderately tolerant, and one F274-2A, that is highly tolerant. To compare that with the group that has 5 tolerant genotypes. And the method is same that we used earlier. And we also think the transcription factor we identify are the potential transcription factors involved in gene regulation in different groups of tolerant rice genotypes under submergence. So this is the promoter architecture of the 5 varieties, that is tolerant, highly tolerant, and extremely tolerant varieties. And these are the transcriptional regulatory modules we identified: MIP, PG, AP2ERF, like many more, ABR1, WORKY, many more. Compared to our previous analysis, here we found more binding sites, because like day by day it's getting advanced. You have more information. The other work we did, like 10 years before this, so we didn't have much information. Since more information available, we can identify many transcription factors this time. And then when we look at the 4 varieties, those are highly tolerant and extremely tolerant, we found they have the binding sites for those transcription factors. Besides that, they have these are extra transcription factors. And when we look at the third one, that is extremely tolerant varieties, those 2 last varieties, we found even extra transcription factor binding sites. And based on all this data, this is the model we developed. If you look at it, these are the 5 tolerant varieties. Nippon bare is moderately tolerant, these 2 highly, and these 2 are extremely tolerant based on their coleoptile length. If you look at that, the initial stage is similar for all of them. What happens when there is imbibition, germination? The ABA level goes down, GA level goes up. It induces the GA responsive transcription factor, and GA responsive, the GA MYB, along with DOF, they induce alpha amylase gene. I didn't put the information here. Alpha amylase gene to break down starch to sugar, and then they get the energy through glycolysis and ethanolic fermentation. And next step, the further elongation. What happens? We found many RAFs. RAFs are ethylene responsive transcription factors. So it shows that the ethylene is important during germination. And we also found many RAF, binding site associated with RAF. RAF always competes with ABA responsive transcription factor because their binding sites are same. So it shows RAF could be inhibiting ABA here. And then we found binding site for EIN3. What happens? It also induces many transcription factors. And the other thing is, it can, if the ethylene level is more, it can, like there will be feedback inhibition, inhibit ethylene. And then there is also involvement of auxin. We know that auxin also helps in elongation. And then, so in all varieties, the initial stage until elongation is same. But then we found some extra transcription factor. Those could be involved in the further elongation. And there we found many transcription factors responsive to brassinosteroid. So those could be involved in the further elongation. And in the two extremely tolerant varieties, we found more binding site. Those are associated with B2, B1M1, and B1M. I cannot see that. Okay, those could be involved in the further elongation of the coleoptile. So our result shows that the MIB regulatory module, earlier when we did 10 years before this, we found both binding site, GA responsive, ABA responsive. Here also we found both ABA responsive, GA responsive transcription factor. And it shows that imbibition and germination, where both hormone ABA and GA play a key role. And then I've already explained DOPE and GAMOIB, they induce the induction of alpha amylase. And we found high enrichment of MIB-box-like element. Those are ABA responsive. And this is the ERF regulatory module. Of course, I have already explained all these things. And this one also I have already explained. The RAF could be competing for the, could be inhibiting the ABA signaling pathway. And in the dark, ethylene plays a double role. We already know it stimulates coleoptile elongation and inhibits root growth. And these modules also interact together. Both hormones also inhibited root elongation. Interaction of both ARF and BGIP could be inhibiting root elongation in rice seedlings, and they could be playing an important role in cell division and regulation of carbohydrate metabolism. And also we found, ABARE and G-box-like elements. Those are again ABA responsive elements, and they could probably be involved in auxin-induced transcription for the longer coleoptile elongation. And interestingly, we found many BHLH transcription factor. We know that they are the major players in phytochrome signal transduction and expression of BR responsive genes. So this BZR1 and PIF1 together with ARF could be inducing genes that are involved in cell expansion, because this coleoptile is a hollow elongated structure. What happens during initial stage, there is cell division, but later it elongates. Because for division, you need energy. And they are underwater, stressed, under oxygen, there is no oxygen. So it's difficult to produce more energy. So that's why they elongate, reach the surface of water to get oxygen. And once they get oxygen, the root and shoot initiates. That is the uniqueness of rice. And we found many BE2, that is again, brassinosteroid responsive. And those could be, also regulate set elongation by brassinosteroid and interacts with auxin responsive factor. And this brassinosteroid is reported to stimulate seed germination and inactivate the negative effect of ABA on seed germination by a negative feedback mechanism that modulating ABA signaling. So like earlier, we thought, how come we are getting ABA responsive genes? Because during germination, ABA level goes down, GA goes up, and we got a lot of criticism. But now, after doing a lot of analysis, I have come to the conclusion that the endogenous ABA level could be acting as an on-off switch, having spatiotemporal regulation to activate or replace other transcription factors and hormonal signaling pathways. Because, there are many pathways, you need ABA for that. They may be, temporarily acting there and not inhibiting the seed germination. So that's the summary I have already explained. We found a complex combination of mechanism that involves different transcription factors. There could be a rebalance between GA and ABA to activate other transcription factor and stress responsive genes. And interestingly, the longer coleoptile elongation in the highly tolerant and extremely tolerant genotypes could be due to the involvement of additional transcription factors such as BHLH and BSBZR, along with racinosteroid signaling. And there could be a crosstalk with other signaling pathways. And these racinosteroid and gibberellic acid, they are known to regulate cell elongation in rice. And racinosteroid regulates cell elongation by controlling also geometabolism. And we found maximum elongation in the two extremely tolerant natural genotypes. That could be due to the additional transcriptional regulatory mechanism that could be governed by transcription factors such as BE2, B1M1, B1M3, and B8. So this variation in coleoptile elongation in different groups of genotypes is due to difference in transcriptional regulation. That's why I wanted to look, because some the coleoptile elongation longer, some shorter. So there could be some involvement of, there could be difference in transcriptional regulation. And after analyzing different varieties, we can clearly see there is a difference in transcriptional regulation, and that could be, that initiates the elongation of the coleoptile. So the variation in coleoptile elongation in different group of genotypes is due to the difference in transcriptional regulatory mechanism that is controlled by specific transcription factor, along with a synergistic crosstalk interaction between different hormones, which needs, of course, further experimental validation, because these are theoretical analysis, but we need experimental validation. And I hope people, slowly, when they do further analysis, hope this hypothesis we have developed could be verified. And then again, I wanted to look at more genes. Those are associated with anaerobic germination. Again, this is from the public data. What they did a genome-wide association study of coleoptile based on coleoptile length, coleoptile diameter in 209 natural rice population. And that was conducted based on SNPs, different SNPs. And the candidate genes they identified are associated with anaerobic germination. So these are the candidate genes: gibberellin 2 beta deoxygenase, then berberine bridge enzyme like 13, protein dehydration induced 19, that's early responsive to dehydration gene, then B3 domain containing protein BP1. So we are quite interested, and this is the phenotypic difference between different variety and the coleoptile elongation. You can see here, just little bit elongation here, much longer. So based on that, they found those genes. We wanted to do some promoter analysis again, look at the promoter architecture, because these are different variety. And we found, interestingly, the same binding sites, more or less same binding sites that we did for the extremely tolerant and highly tolerant varieties. But besides that, we found some extra binding sites. Those are MIB3R, which we did not find in any of those varieties, those genotypes. And then TSO1, TCX2, TCX3, SHR1, WRL1, and AHL. So then we develop a model based on the promoter architecture analysis. It's exactly the same that I explained for highly tolerant and extremely tolerant varieties. Only difference, we found some extra transcription factor. Those could be involved in the epigenetic modification. So these are the new transcription factor binding sites we found in those candidate genes. So these transcription factors belong to repressor type MIBs. Earlier, we are finding GA responsive and ABA responsive. Now we have found some transcription factor. Those are repressor type, but then they mainly suppress post-mitotic cell during organ development. So in our case, the coleoptile is just an elongation. There is no cell division, like further thing. There is no differentiation. So that could be, controlling the cell elongation, cell division there. And also, it plays a role in inhibiting cell division in response to DNA damage. So we believe that that could be also playing a role. And then both this MIB3R1 and 3R5 could be involved in repressing organ development, that is root and shoot, to support the elongation of coleoptile to reach the surface of water for oxygen. And these transcription factor together with these are also E2F, E2FC, these are again repressor type transcription factor. They could be potentially limiting the mitotic gene expression in elongated coleoptile, because initially there is cell division, then it stops. And these are the new transcription factor. As I said, it's just the cell elongation, and it's known that cell division is active for the first days of submergence, and then there is cell elongation as this process consumes more energy than cellular expansion. And these transcription factors possibly be involved in the inhibition of cell division to accelerate cell elongation of the coleoptile. And then these transcription factors, they induce genes in the glycolytic pathway and fatty acid biosynthesis. And in rice, two WRI1 genes have been identified, but their role in the seed germination is not known yet. And this one is HRR. We found the binding site for SHR1. This is involved in the epigenetic control of gene expression, histone modification, and also methylation. So this could be involved in the control of cell division, and its expression is regulated by E2FA and E2FB. That could control the G1 to S phase transition of the mitotic cycle. So all these candidate anaerobic germination genes, they possess binding sites for both SHR1 and also these transcription factors. These are the repressor type. Hence, there is possibility that these two transcription factors could cooperatively be involved in epigenetic control of gene expression during the elongation of coleoptile under flooding condition. And this transcription factor is AHL. This one is involved in lipid mobilization and fatty acid beta oxidation, and that interacts with lipid mediator phosphatidic acid and degrades lipids to provide acetyl-CoA, substrate for the glycolysis cycle to produce glucose during seed germination and seedling establishment. And the comparative analysis between rice seeds germinated under air and anoxia identified 28 transcripts of proteins associated with lipid metabolism. So it does mean that it could be playing a role there. So AHL might be controlling, this one is different. Okay. Okay, this one. Might be controlling, then we found another transcription factor regulatory module. I have already discussed. But lipid metabolism is more towards biosynthesis in rice seeds germinated under anoxia hypoxia, as ATP produced during this condition is involved for the maintenance of membrane integrity. So it shows since lipid metabolism helps to maintain membrane integrity under anoxia hypoxia, this transcription factor regulatory module could be controlling the detoxification of ROS in coleoptile during anaerobic germination. I have already explained that we found very high enrichment of ABA responsive transcription factors, and that could be due to the pre-existing ABA and maybe involved at the beginning and later stage of germination. in a spatiotemporal manner through activation and inhibition machinery. And if you look at the candidate genes, that is OSBP1 and then the other genes. The authors, they reported that the ABA content was fluctuating at a low level during flooding condition. And this gene could be involved in fine-tuning the ABA signaling pathway to induce seed germination. And then they did the quantitative, the measurement for ABA. What they have found that there was a gradual decrease in ABA content in the first 3 days after flooding, but then increased on the fourth day. So it makes sense because, as I said, they may be acting as a spatiotemporal manner. They never required, they're activated, again they're inhibited. They're activated to induce other transcription factors to support seed germination. And then this gene that is early responsive to dehydration is a gene related to drought stress. It encodes dehydration-induced DIL9 protein. This gene could be directly or indirectly inducing ABA response genes by altering ABA content to regulate the phenotype of the coleoptile. So all our data, we have identified, we have analyzed the promoter architecture of many genotypes, and looking at them, it shows that ABA level could be acting as an on-off switch, regulating in a spatiotemporal manner to activate, replace other transcription factors and hormonal signaling pathways. And these transcription factors, of course, involved in the more elongation of the coleoptile. And there is coordination of both auxin and BR response regulation. And these transcription factors could be involved in epigenetic regulation of coleoptile elongation. And then to compare these data, we did we look at the upregulated genes in the aerobic condition. And what happens when we look at the promoter architecture, we found either less the ABA responsive to, transcription factor binding or cis elements are less enriched or absent. And that implies that the importance of GA-dependent initiation and germination of rice seeds in aerobic condition. And besides, we found many other transcription factor binding site related to epigenetic regulation and racinosteroid responsive genes. We didn't find any of those binding sites in the genes. Those are upregulated in aerobic condition. And that's the summary I already explained. And I would like to thank NUS Environmental Research Institute, National University of Singapore, also Department of Chemical Biochemical Engineering, National University of Singapore, because I was there and we did all the modeling work there. And I would like to thank also Next Generation BioGreen program, because we got the grant from there to work on this rice work. Professor Dongyopli, he is a chemical engineer. He was, guiding us for the rice metabolic pathway. And Meyapan Lakshmanan was a PhD student. So when I was in chemical engineering, he got his PhD on rice and he developed the model. And it was really exciting, like chemical engineers working on rice and developing the model. I was helping them with the biology. That was really satisfying. And then we have a collaborator from Texas Tech, Professor Reyes. He also helps with analysis, comment, and things like that. Thank you so much for listening.